Hello, lovely biology students. Ms. Steensen here. We are starting our unit on cellular informatics and division, and the goal is to finish up this unit before spring break. For that reason, we're going to be doing some stuff online as well as in class to make sure we get the most from our learning. This first lecture is about the structure of DNA. We're also going to look a little bit at RNA. By the end of this, you should be able to describe the structure of DNA and relate the structure to its function. Remember, one of our big ideas in biology is this relationship, structure, and function. There's also this guiding question: what is the role of DNA in coding for the functions of cells? Keep this in the back of your mind because by the end of this, we really want to be able to answer that. So remember, our big function of DNA is to store and transfer genetic information. It really is what makes you you, and that makes it pretty great. So nucleic acids are one of our macromolecules. Remember from earlier in the year, we had four big types of macromolecules. We have our lipids, our carbohydrates, our proteins, and our nucleic acids. Like all macromolecules, there is a monomer, a small section, that gets repeated over and over and over again in order to make our polymer. For nucleic acids, the monomers are called nucleotides. We're going to look at two different types of nucleotides, the one for DNA and the one for RNA. They're really similar, but have a couple important differences. Both of them are going to have three big parts: a phosphate group, a sugar, and nitrogenous base. So this picture over here we got our phosphate group, the sugar, and nitrogenous base. For DNA, you're going to have deoxyribose as the sugar, and adenine, thymine, guanine, and cytosine as your possible nitrogenous bases. For RNA, you've got ribose as the sugar, and adenine, uracil, guanine, cytosine as the possible nitrogenous bases. I tried to highlight the differences here in red so you can see those really easily. Looking at these pictures, this one down here is RNA. If we look here, there's these little numbers. Those are numbering the carbons. So at the tips of all of these, there is a carbon there. So there's a carbon here, 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 and here. Both of these have 5 carbons. The big difference between these sugars is if you look off this carbon, there's this OH, this hydroxide group, and if we look off this one right here, there's just an H. So deoxy means like less oxygen, or less oxygen. So we can see there's one less oxygen right there. So if we take a bunch of different nucleotides and put them together, that's where our code is stored. The name of our polymer is nucleic acids. On the outside of this polymer, there's going to be a sugar phosphate backbone. What happens is a phosphate group and a sugar group come together and create a covalent bond. So remember, covalent bonds are where electrons are being shared. That makes them really, really strong bonds. In the center, we've got some really, really weak bonds. These are all hydrogen bonds, and hydrogens are just interactions between slightly polar bonds. So these parts, really weak. These bonds on the outside, really, really strong. In the middle, though, the order of these nitrogenous bases is really where we get the storage and transfer of this genetic information. Adenines and thymines, they're always going to bond together, and between them, there's going to be 2 hydrogen bonds. Notice these dotted lines, those represent the hydrogen bonds. On the bottom here, guanine and cytosine, they have 3 hydrogen bonds holding them together, and they're always going to bond. The 2 halves of our DNA look really similar, but there is a little bit of directionality. Notice here we've got this 5' end labeled, 3' end down here, versus the 5' end on this side, 3' end down here. What that means is, see how there's sort of the phosphate group sticking off one side, the sugar sticking off the other side? It makes them sort of unbalanced. The phosphate group, it's always going to stick off the 5th carbon if we start numbering at the oxygen, so we call that the 5' end. Versus the other end here comes off the 3rd carbon, so we call that the 3' end. Notice the 5 and the 3 are sort of opposite each other in the 2 different halves. That's why we call DNA anti-parallel, because these are parallel to each other but flipped around, hence anti-parallel. When we get to our nitrogenous bases, there's 2 different categories. There's purines, and they're going to have 2 rings in their bases. So if we look down here, our purines, we have adenine and guanine. They have 1 2 different rings. Pyrimidines, on the other hand, only have 1 ring. So we've got uracil, that one just exists in RNA, thymine, which only exists in DNA, and then cytosine, which exists in both, and those all just have 1 ring. Base pairs, they're always going to form between 1 purine and 1 pyrimidine because that keeps the width of the DNA equal all the way through. Again, thymine and adenine, they're going to create 2 hydrogen bonds between them, and cytosine and guanine are going to create 3 hydrogen bonds between them. So this animation sort of shows the process of creating a DNA. Notice that as these come in, phosphorylation happens. That's just phosphate group coming off in order to form that covalent bond. That creates our sugar phosphate backbone, which is really, really strong. Notice which type of nucleotide is coming in is dependent on which things it can pair with. T is always going to pair with A, G with C, A with T, C with G. Those are always going to be the groups they form. So we notice which one comes in is because of that, and then creating this covalent bond on the outside. Again, we see our 5' end, our 3' end, and those are anti-parallel. So after the DNA has formed its 2 different sides, it eventually is going to wind into this double helix shape. This is just because of some attractions between some slightly negative and slightly positive parts of the DNA, and this creates what we think of as DNA. So we really have a lot of DNA. It's estimated that in every single one of our cells, we have enough DNA that if we just laid it out straight, it would be something like 2 meters long, which is, taller than us, which is crazy to think of because we have 50 trillion cells. So that DNA has to be really, 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 really tightly packaged. The process of packaging up that DNA is a little different between prokaryotes like bacteria and between eukaryotes like us. Prokaryotes just have a little bit of DNA, so theirs doesn't have to be super, super wound up. But as eukaryotes, our DNA has to be really, really tightly wound. When we say that eukaryotic DNA is associated with proteins, this is what we mean. Down here at the bottom of this image, this is the least tightly wound. This is just straight up DNA with its double helix. The first step in getting it really tightly packaged involves some proteins. They look kind of like little beads here, but those are supposed to represent histones, the name for a type of protein. They act kind of like a spool that you wind thread around, but you wind that DNA around it. And then all of those histones are able to come together and wind around themselves again. When the DNA is all wrapped around itself again here, we call that a solenoid. It's just getting the DNA tighter and tighter and tighter. It then is going to supercoil, meaning it's going to wrap around itself again. It's eventually going to form something that looks a little bit like this. This is what we call chromatin. This is how DNA is most of the time in our cells. It's got these super, super tightly coiled sections, and in the middle, it's got a lot of protein holding it together. All of these guys out here are called looped domains. And again, it's this combination of protein holding stuff close together in addition to that DNA. When it comes time to create a chromosome, when it comes time to divide the DNA, this stuff is going to get even tighter wound, and eventually it's going to be so tightly packed that it looks like the chromosomes we think of seeing like under a microscope. All of this stuff is super, super tightly wound just so it can fit within a cell. Here's another way to think of the different winding process. Down here we have the width of this in nanometers. Remember, nanometers is 10 to the negative 9 meters, so super, super small. Here we've got a single piece of DNA. Then we wrap these things around the proteins. It gets a little wider. Then we wrap those around themselves again, and we get a little wider. And then again, and wider, 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 up until we get to the chromosome. When mitosis is ready to happen, see how much fatter this stuff is than it was to begin with. That's just so it all stays together so it can be easily separated out. So whenever it's not all tightly wound like that, ready to be separated, the DNA is constantly in the process of being copied, either to make more DNA or to make RNA and eventually make proteins, which is what makes us us. I want us to understand some big differences between DNA and RNA. They're both nucleic acids. We're going to see them at different places in the processes we're going to learn about next. Whenever we're comparing something using our IB command terms, that really means compare and contrast. We need to say how each thing is either similar or different. So I try to do that in this slide. Here we see DNA has 5 carbon sugar, so does RNA. But in DNA, that one is deoxyribose, like less oxygen, deoxyribose. In RNA, it's just ribose. In DNA, we've got 4 nitrogenous bases, same with an RNA. But in DNA, there's cytosine, guanine, adenine, and thymine. C and G are going to form a bond, A and T are going to form a bond. In RNA, we've got cytosine, guanine, adenine, and uracil. C and G are going to form a bond, and A and U are going to form a bond. And then in DNA, it's always going to be that anti-parallel double-stranded molecule, versus in RNA, it's going to be single-stranded a lot more often. So keep these ideas in the back of your mind as we go forward and learn about a lot of the different processes that allow us to copy DNA, make proteins, and make us us. I want you to use the assessment statements to review and study to be able to complete all the assignments that we have coming up. Please come to class ready with any questions you have about this stuff, and have a lovely evening.